So we want you all to do well in today's lab. So I have a little video of a demonstration to help you out. First, if you look at your desk, you'll notice that you have a tray. It has different samples in it. You have your control samples labeled blood type A, blood type B, AB, and O. You also have your patient samples labeled patient 1, 2, 3, and 4. You'll notice the 4 tubes in the middle are labeled with a strange alpha, or it kind of looks like a fish symbol, and an A or a B. Those are going to be the antiserum samples that we talked about earlier. You'll also notice if you look on your desk, you have different petri dishes. Some dishes are labeled patient plates, others controls, and some dishes are labeled anti-A and some anti-B. All of the dishes are dissected into quadrants. And what we're going to do is we're going to show you guys how to use these plates to complete your experiment. So the first thing you need to do is you'll need to get your yellow pipette and add 25 microliters of the appropriate sample into each quadrant. So in today's lab, or in today's demo, this iodine is going to be my blood sample. So this will be blood sample A. So the first thing you have to do is take 25 microliters of blood sample A and place it into quadrant A. And get a new tip. You don't want to mix your blood samples because that could lead to skewed results. Next, you'll add 25 microliters of blood sample B into quadrant B. Again, getting a new tip after the addition of every sample. Blood type AB into quadrant AB. New tip. And finally, blood type O into quadrant O. Next, you'll need to add the appropriate antiserum. If you look at the plate, this plate is labeled antiserum A. It's going to be represented by our bromophenol blue. You'll use the gray pipettes to add 10 microliters of antiserum A to each quadrant. So we'll add 10 microliters of antiserum A to quadrant A. And we'll stir using the pipette tip and get a new tip. Make sure you always get a new tip after every sample. Remember, you can contaminate and skew the result. Next, we'll add 10 microliters of antiserum A to quadrant B and stir. 10 microliters of antiserum A to quadrant AB and stir. And finally, 10 microliters of antiserum A into quadrant O and stir. Based on the results of the reactions, we'll be able to tell, using the two plates, what type of blood each patient has.